so again, the ribosomes, they're made of rRNA, ribosomal RNA and protein, and the rRNA was made by the nucleolus. They are not one piece, they are actually two subunits, a large subunit and a small subunit. You can see it in the picture down here at the bottom. Their job is to make proteins, but again, nothing's actually happening inside of a ribosome. What's actually happening is the large subunit and the small subunit work sort of like a paperclip to hold mRNA in place, and then the mRNA is read and proteins are assembled. So their job that they're doing is something that's sort of external, not internal. Some are just floating around. Those are called free ribosomes. And then there's also ribosomes in the cell that are around the rough ER, and those are actually called bound ribosomes. All of them make proteins, though. That's their job. And really, they're not necessarily synthesizing them. They're more of assisting in the assembly of proteins is probably a better way to word it. We'll do an entire chapter about protein synthesis much later in the year after Christmas. So we'll talk more about the details of it. But that's the bottom line. They're not actually just a single dot. They're two pieces. And then in between those two pieces, they sort of hold stuff in place for the assembly of proteins. All right, now, I know this is not in the outline, but I wanted to explain this. All these cell parts that we're going to be talking about, the nuclear envelope, the membrane, the ER, the Golgi apparatus, lysosomes, vacuoles, and the cell membrane are all part of what we call the endomembrane system. So what I want to explain to you is this. If you look at a cell, all of these are surrounded by membranes. The nucleus literally is connected to the rough ER. There's literally attachment points. And then the rough ER, when it might make a product, let's just say, make it in green. So let's say the rough ER makes some kind of product, and it wants to send that product somewhere. What'll actually happen is a piece of the rough ER membrane will break off and travel, let's say it goes to the Golgi bodies, and that piece of membrane will then fuse with the Golgi bodies and become part of them. And when the Golgi bodies are going to send something out of the cell, a little piece will break off, travel to the membrane, and just like literally like this, it'll basically fuse with the membrane, and that product would then be released. And now this would be part of the cell membrane. So when we call this the endomembrane system, it's literally because the cell membrane is not the only place we find membrane. The pieces of cell membrane end up on the Golgi bodies, they end up on these little vesicles and vacuoles, they end up on the ER, they end up fused with the nucleus. They are breaking off and fusing with other areas all over the cell. All of these areas are surrounded by membranes. So they call it the endomembrane system because pieces are sort of breaking off and moving around throughout. All right, so the only cell part we're going to talk about today that's part of the endomembrane system is the ER. So the ER, like I said, it starts right at the nucleus. There's actually areas where it's attached. I'll show you on the next slide. And there's two kinds, which everybody already knows, the rough and the smooth. The rough is the one that has the ribosomes on it. The smooth does not. Here's a picture. Would this be rough or smooth in the picture? Good, rough. Very obvious. I don't know what kind of stains they use, but it's very, very obvious. Those maroon dots would be the ribosomes. And then inside that yellow is the actual endoplasmic reticulum. And probably over here in blue on the left side is probably the nucleus. I'm not sure. On the right, it looks almost like that might be a mitochondria or something in purple. It doesn't matter. You wouldn't have to identify it from a picture. But that sort of gives you an idea of what it looks like. This is another electron microscope picture of it, which to me is not as pretty as the first one. Oops. But I did want to show you this if I make this a little bigger. So see what I mean? Here's where the rough ER right here on the left side is actually fusing here on the right, and here on the left is actually fusing with the nucleus. So it's these big, long canals that form a system of membranes, and it's almost like a maze. Like you could literally go in here, and then something could break off and go to this one, and it could travel, and end up in this one and travel this way. So it's a big series of canals, and stuff can sort of travel throughout the cell. And when pieces break off, they break off into these little vesicles, piece of membrane, everything's sort of captured in a little ball, and that'll go fuse with another cell part. Here's the functions. I probably shouldn't have had it all up here at one time, but the smooth, most people know the rough ER. Most people know the rough ER modifies proteins, which makes sense, because the ribosomes are made of protein, and the ribosomes make protein, and then right there is the rough ER. Smoothie ER people are a little less familiar with. So what does it do? Number one, it makes lipids and steroids. So remember steroids from the chapter we just talked about. So it would take in cholesterol and then make the steroids you need, maybe estrogen or testosterone or cortisone or whatever. The smoothie ER would be in charge of doing that. It can help with metabolizing carbohydrates, meaning breaking them down. A really big thing, probably one of the main things that the smoothie ER does, is detoxifying poisons and drugs and things. When you eat, and the first thing that's going to happen when you absorb all the nutrients from your food is it's going to go into your bloodstream and it's going to go to your liver. Your liver is then going to, the cells of your liver have lots and lots of smoothie ER. And that smoothie ER is going to start processing anything in that meal that needs to be broken down. So, for example, you take an antibiotic. You might say, why couldn't I just take one antibiotic, like a super one, that would just last me the whole 7 days? Why do I have to take one every 12 hours for X number of days? Because once it gets in your bloodstream, it immediately goes to your liver, and your liver immediately starts deactivating it and breaking it down and processing it, and then you're basically just going to go to the bathroom and get rid of the remainder of it. And when somebody drinks alcohol, if they're drinking it really slowly, they're not going to get much of an effect from it because it's going to go to their liver, and their smoothie ER is going to immediately start breaking it down. It basically just processes the alcohol into sugars. Is all it's going to do. But if somebody drinks a 6-pack of beer on an empty stomach in a 10-minute period, they're probably going to be sick because there's not going to be enough smoothie ER to process all of it. So that's when it's going to stay in the bloodstream, and the person's going to have whatever side effects they're going to have. You know, they're going to get dizzy or stagger or whatever, and maybe throw up. Like I said, if it's too much, then that's your body's way of saying, I got to get this out of me any way I can. But anything you take, so even if you're taking Tylenol, it's going to get processed. Some things process faster than others. The other thing that it does is it stores calcium ions. You need calcium for muscle contraction, so that's an important ion, and it can be stored in the smoothie ER and then used as needed. Again, the rough ER you already know. Most people usually know what it does, but now that you understand the levels of structure of protein, it might make more sense what's happening here. So remember, primary structure happens at the ribosomes. That's when the protein is actually built. So the amino acids are put in a particular order. But remember what happens after that. You get the secondary and the tertiary structures and sometimes quaternary structures. A lot of that happens in the rough ER. So the protein is made, it goes into the rough ER, and now it finishes folding up. It might get cut, extra polypeptides might get added to it, and all the final stuff will happen. And then that protein a piece of membrane will break off, that's a vesicle, and carry it to some other location in the cell where other stuff might happen. It also makes glycoproteins. I mentioned those on the previous slide a few minutes ago. Those are proteins that have carbohydrates attached to them. So they had it drawn like this, and it looked like it had like little hexagons coming off of it. So those are ID markers, which are really, really important for identifying cells as belonging to you. Like your blood type, or even just general things that tell your immune system not to attack your own body. So that's what glycoproteins are. All right, and that's actually where we're going to stop today.